I'm recording the session. Welcome to the webinar of HeFarm. This webinar on 18th of March of 2026 will be split into two sessions. Well, this is the first webinar. The first webinar will have three presentations from 11 to 11:20, from 11:20 to 11:40, and from 11:40 to 12, 20 minutes each. And then after this, the second webinar will be also today in a second session with from 12:30 to 1:30. Okay, with three sessions from Antonia Camí, Diana Yorga, and Sonia Piro. So we will start with my presentation in order to be in time. And my presentation will be about dispersion and transport of microorganisms and the relevance for biosecurity. This, as you know, is a webinar based on the results of the Horizon Europe program, the project HeFarm, that engage all these partners from different countries and different expertises. It engaged the universities, research centers, companies, even associations like Catal from the Lombardy or the Association for Standardization for Romania at Tisasmo. All this scientific and technological team has been engaged and involved in the development of all the results that will be exposed in this webinar, the webinar that will be later, and also the all other two webinars that are also announced in LinkedIn. The project had a duration of 3.3 years and a budget of 5 million, and also had many other people cooperating that we have to thank and that we want to acknowledge. We don't have time to go into details, but we have from the IMIDRA, from Community of Madrid, different farms that have cooperated from different sites, starting from the Extremadura farms to Green Pork and Calderones, and also Avícola Barco, where different devices were tested. Also, the Association of Selected Cattle from Spain, and the Association for the Growth of Snails. Additionally, from University of Barcelona, and also from even from the military emergency unit, that you will see some of the images that we will share. In HeFarm, the philosophy or the leitmotif that we have is that we have a lot of different pathogens. These pathogens may be transported by only a few means of transport. So if the number of pathogens may be thousands, it's impossible to create vaccines and specific means for, let's say, immune means and so on for each of them. They are not available, in fact. So the point is that if we act as a whole on all the bunch of different microorganisms that may be coming, then we will have a biosecure or biosafe facility. So we identify the different routes or different means of transport and address each of them, particularly focusing on air and aerosols that were the main forgotten, or usually they have been those that well, have not considered usually in the biosecurity studies. Also, not only incoming transmission, but also outgoing transmission is important. In this case, it changes, for instance, water changes for liquid waste, and so on. Sorry. How we assessed biosecurity? We wanted to assess in a numerical and quantitative way. So, for instance, we have first experimental methods and theoretical or computational models. In experimental methods, we use stress tests. That means that we can measure how many microorganisms are transmitted. For instance, how many are dragged by boots when you walk into a farm, or for instance, in a toilet, how many aerosols, how many microorganisms will escape, for instance, from the sewage into the toilet room through the siphon that you have here. That is something that numerically we can stress and measure. In the same way, we can also make some step tests. Step test means that we can disinfect, in this case, a railway car, we can disinfect it, and then measure how the microorganisms are coming back, because that will show what are the routes that they are using to recolonize the environment that has been cleared at a certain time. It's like in other engineering disciplines, it's called a step test, and it's usually it's useful for determining the behavior of a system, a dynamic system. Finally, we have fluid mechanics, Brownian motion, a combination that can be used in computational models in order to predict what is the behavior, what are the transmission coefficients for particles of a particular size. This is an example of how Brownian motion is important. Let's say it's not just the flow of air that drags the microorganisms, but the Brownian motion for a micron size particle that acts at this scale. You remember that it's Albert Einstein was awarded the Nobel Prize, not for the relativity, but for the Brownian motion description. You can see that just by Brownian motion, the particles, the airborne particles, are disseminated in the air. In fact, when Pasteur made his experiment, he provided their glass bottles, he provided them with a long capillary, narrow, that prevents Brownian motion through it. Let's say you have Brownian motion of microorganisms here in the air, it's extremely unlike to them to penetrate into the glass. So let's say that if he hadn't used this long pipe, probably if he had just an open mouth here or here, just Brownian motion would have spoiled his experiment, and probably we would be now believing in spontaneous generation and this kind of things. So this is, I think, quite relevant. So our question is, how can a microorganism travel from an animal to another animal if the distance compared to his size is as if we want to travel from Madrid to Paris in just a single jump? It's similar. It's something scalable. So we have to focus on how the aerosols behave. You know, the aerosols are usually, you see here, Madrid in a polluted time, and how aerosols make even dark the air. But if they make dark the air, it's because they are bigger than the visible light wavelength. But even there are other aerosols that are even smaller, and you cannot see them. Okay. Well, mechanisms for aerosolization are many. You can have heating, creating, well, all the bubbling, busting creates aerosols, for sure smoking, but also airflow on a surface, on a wet surface, on a water surface, will create aerosols. But also we have skin. Skin is growing from the bottom to the outer part, and is continuously ejecting skin particles. This is a powerful aerosolization mechanism, as it has been shown in our experiments. You have to compare the normal skin tissue with the lung tissue. You have, for instance, in the trachea, this is for mouse skin and mouse trachea. You can see how in the respiratory system the thickness of the skin is thinner and thinner as long as you penetrate into the lung. Also, coughing is another mechanism that we know that generates aerosols, but also just breathing may also be a mechanism for generating aerosols. So going fast, let's say that you have to consider all the aerosol dynamics that is governed by the Stokes law. Is a particle of the size of a micron is like, or under micron, is like behaving in a jam, in a viscous media, and they will fall at a very slow rate, governed by this Stokes law. And that means that you have a particle at 2.4 meters of height. It will take, let's say, for a 10 micron, 13-14 minutes to fall down. But for a 2 micron, it will be hours or even days. Also, dragging of particles is governed by their size. This is just for 1 micron. You will see that will follow essentially the lateral flow of 1 meter per second. So essentially, it will follow the air flow. And also another rule is that it will penetrate deeper for smaller particles. So the smaller they are, the deeper they will enter into the lung. That will create a problem, that is that if something reaches the aerosols, it will really very difficult to extract from the aerosols. And that makes respiratory agents to be much more risky, riskier than the, let's say, any other pathogen or microorganism. Well, during the FP7 Counterflow project, we developed technology for collapsing aerosols from air. And this technology was demonstrated with spores of a surrogate of Bacillus thuringiensis. You can see how in a few minutes we can remove them from air. And also here you have a video that was made in cooperation with the military emergency unit. And you can see how the jets of fog that we create absorb any aerosol, like this red smoke, and collapse it onto the ground or to the site where you are projecting that. So this technology can be used in a twofold way. It can be used for sampling aerosols. You have some pictures of the device working in a farm, and also of the new device that has been evolved during the project that works with batteries and is completely portable. Here the system generates a jet of fog that absorbs any aerosol that is around it in the environment and brings it to this container, this plastic container, where you will have your sample, your liquid sample, with a suspension of all the microorganisms that have been dragged into it. Well, these are some examples of some of the microorganisms. For instance, the Pérez virus, is the pig reproductive and respiratory syndrome virus that was detected in some farms. But also, if we made metagenomics from these samples taken in farms, we found thousands of different species of bacteria, of fungi, of viruses. So essentially, they are the air is full of these microorganisms, and air or aerosols are a common means of transport for let's say microorganisms that you never expected that they were here. Obviously, we published this in a paper that you can find in the website. I think we have we have it referred. And the point is that in this test, it was demonstrated that air was filled of these microorganisms, but also that skin ejection was the main mechanism in the aerosolization of all these microorganisms. Also, this is for a chicken farm in Poland. You can see again the sample, the taking sample. This is the metagenomic results. Also, it was shown interesting effect, and this was presented in a conference in Italy last year, is that the composition of the aerosols at different heights is different. So that means that there are some species that are more abundant in the higher parts than in the lower parts of or layers of air. And that means that we can measure how easily they aerosolize or how volatile they are. This repeats again in a snail farm, and that is a demonstration that it's not that chicken wings are well, move the air, or pigs, calves, or I mean, breathe strongly. These quiet and calm animals that are snails, they also are quite effective machines of aerosolization. The interesting results in these snail farms, that was the CIOP snail farm in Igualada, close to Barcelona in Spain. The interesting thing is that, well, the presence of animals was associated with the presence of the bacteria in air. So it was clearly associated to the presence of the animals. These are some of the results of the abundances in the metagenomics analysis. And we can find here how, after decontamination, obviously the relative proportion of the microorganisms change, but also their absolute number. Let's say there is a clearance or a removal of the microorganisms that are aerosols. Well, I'm sorry that I have to move quickly in order to be in time and not to input any delay. But let's say to show how this kind of measurement of the aerosols or bioaerosols can be important. Let's say that something that has been proved is that samples at 150 meters away from a farm that was positive in avian flu, well, 150 meters away, then we found avian flu viruses. Well, we found the let's say the detection of the genetic material. It was not proved that they were active, but it can be inferred that it was quite likely active viruses. So it opens, let's say, essentially these results. What demonstrate is that air is full of these microorganisms, usually invisible to the naked eye because they are smaller than the wavelength of light, visible light, but that they are really present, and then aerosolization and de-aerosolization mechanisms are really quite effective in working. Sorry that I forgot to translate these conclusions. So let's say that aerosolization is a general phenomenon. And obviously, if you want some biosecurity and biosafety implemented and effectively used in a site, you have to consider how to prevent aerosols to come in or to go out. Eventually, we have validated tools, effective tools to study aerobiology including volatility or easiness to aerosolize, and how, let's say, you have a climatic system or a climate heating or ventilation air conditioning system, you have to take into account that the air is a reservoir for the microorganisms. So that's it. I think I'm just in time, but if anybody has any question, a quick question, we may answer it. If somebody wants to ask, I think you may raise hand or you may activate your microphone. Okay, if there is no question, then we may go into our next session that will be by Tekla Engelhardt from the Veterinary University of Budapest. And let me, Tekla, make you...
Hello, good morning.
Good morning, Tekla.
I just tried to share my screen.
You can share it now.
Share content, okay.
Yeah, you can share it.
Just a few minutes because I have to allow everything. Oh, okay. I cannot share it. Can you share my, because I have to open my...
I think Christina, do
yes,
you have the presentation?
Yes. Sorry for that.
Yes, no problem.
Yeah, thank you. So I will present the results from the Equine Clinic. We had some hygienic issue with the clinic. I mean, they have lots of problem how they can make the disinfection and the cleaning, or maybe not the cleaning, but the disinfection is really in an effective and proper way. And that is what I will present for you today. So you can change to the next slide, please. So I think you already know it very well. Under the Have project, our task was to try to identify how can we improve the biosecurity and how can we identify the main microbial pathways of transmission on farms or any other clinics or any other places which are linked to the animals, and how can we improve the biosecurity, the one health, the safety of the consumers, and so on. So for that, we measured several things, and we identified the cleaning and the disinfection will be crucial, and we have to somehow control the microbial transmission, and that is what I will present in that presentation. So you can move forward for my next slide. It was just a short reminder, because originally we did identify several transmission routes, and we focused on the other materials and fomitids on the clinic, because we realized how important are the surfaces in link to the biosecurity. And if our surfaces are under control, our equipments are under control, the biosecurity will increase. And we also identified some really critical surfaces which are absorbent and has a porous surface, and can the microbes attach really well into that surfaces, and the cleaning and the disinfection are really hard in a normal way, but they are regularly used to it. And we also identified some other really critical points like feeders, the drinkers, the nose switches, and of course they have lots of manure force showers and etc. So for the effective cleaning and disinfection, protocols and tools are really required at that certain point in 2 years ago or something like that. You can see a picture about the stall, and you can already, I think, recognize the floor. It's really hard to clean and make a disinfection. We also have lots of wooden material and concrete material as well, and you also can recognize the counterfog equipment that we used it. So the next slide, please. And maybe you can play the video, because I have a video how they use the, yeah, that's great. So we use the counterfog because it's a recently developed equipment, and it's a really fast application of nanometric droplets of the biocides, and that was the key point here because of the really complicated surfaces. And it also seems it will be prevent the resuspension of the pathogens, and as you can see, it's really easy to use. And I think that is, of course, it's really important how effective the apparatus, the equipment, how effective the biocides that you use, but it's also really important how easy to use for the workers, because if it's some kind of complicated equipment, they will reject to use it because they will choose that kind of equipment which are easy to use, and the setup is just a few minutes and so on, and it's really easy to use it during the disinfection, and they actually really like it nowadays. So please move to the next slide. Maybe you have to click on the, yeah, thank you. So what we performed and what was done, we used it at a practical application, the part one, the testing part, when we just select some stall with the selected stable, and they were thoroughly disinfected. And after, the samples were taken from different locations before and after the process, of course, because you would like to compare how was it efficient or how wasn't it efficient. And it's also really important, we made that disinfection, and after we wait for 48 hours after the disinfection in order to obtain a more realistic picture of its effectiveness. And how they perform the sampling, we use the swabs, and we always perform samples from the 100 cubic centimeter surface from different location, and after we inoculated onto half concentrated tryptic soy broth, and we incubated it for 24 hours at 30 degrees to get a picture about the total colony count. We didn't interested how it's effective for a certain pathogens at that certain time. We just would like to see in general how it's effective, because in a first look we don't know what kind of pathogenic microbes is worth to sample and try to detect. It's much more easier to see the total colony count and see how can they decrease after the disinfection by the counterfog. So the next slide, please. So here is the results, and you can see it was a really effective procedure. the log n is represent the total colony count, and you can see the different places. We make samples from a drinker, the feeder, the steel grid, the door, the concrete pillar, and the window. Usually we always make a sampling like this, so that is the surfaces what we usually check, because it's a really nice, give us a really nice picture. You may already maybe recognize the door. It wasn't any colony count before the disinfection, because the door I usually make the disinfections when they working at the daily routine, of course, because they wash their hands, make a disinfection on their hands and so on. So that is the reason. And of course it was always after the cleaning, but before the disinfection when we took the samples. And you can see we have when we perform the disinfections, we could eliminate all of the bacterial number. So we can say it was a really effective procedure. So after that, the next slide, please, Christina. So after that, the workers used that apparatus for a long run. They started to use their daily routine, not the daily routine, but the disinfection routine, let's say. And they have every 3rd month, one stable was fully cleaned. They will move out all of the horses, and one stable, they have 4. And every 3rd month they fully cleaned one stable and disinfected by the counter fog. And of course, within that 3rd month, they also perform, of course, the cleaning and the disinfection. But in every 3rd month, let's say they have that huge cleaning and disinfection procedure to ensure the biosecurity and the safety. And in that case, the samples again were taken and monitored by the microtester apparatus as before. And again, we always measure the total plate count in the half concentrated TSB broth at 30 degrees for 24 hours. So we will receive a really fast results. And we discussed regularly the results with the staff, because of course it's the people are working there, and in some cases they miss some surface, or they skip some part of the stable, and that was really well recognized after the results. So the next slide, please. So the blue lines represent the before results, and the green one the after the results. And when you could recognize just almost nothing, it means it was under the detection criteria. And what we saw from for the long run results, when they made that cleaning at least every 3rd month, but usually they performed it most frequently, but just for some of the boxes for the horse. So what we can see, the counter is in a really effective apparatus, because mainly they perform it well, and we cannot detect any bacteria. But in some cases, we could recognize some colony counts, or even it wasn't some, because it was around the 5 log or 4 log. And we always go back and check it what happened. So for example, it's a really nice example for the concrete pillar, because the concrete pillar surfaces are really absorbent, and they can keep the microbes attached really well. So they make a cleaning again and the disinfection again, and after they learned it, they have to pay attention on the concrete pillars. And after the next times, it was the results was really nice, and it wasn't any problem about the pillars. And we for example, it was last time in January when they, maybe that wasn't that slide, but it was also the results from January, because they thought I forgot to clean the feeders, and it was also was from the results. And when we checked again, it was obvious they forgot to clean and disinfect the feeders, so they made again. So it was also demonstrated how important to discuss the results with the workers and make communication with them. And of course, they focused on the previous problems like pillars, the concrete pillars, the drinkers was also a problem before, but after they just forget the feeders and they went back, and of course they clean it and make a disinfection again, and it was fine after that. So, and it was also it was really great to see how the workers accepted that technique, and they really liked it to use because it's really easy. And but keep the communication with them, it's really essential. So the next slide, please. After that, in parallel, in the end, we also performed a much more deep results, because we were really curious if it's always some surfaces when some bacteria could survive, but if they could survive, because in the results we could detect 1 log or 1.5 log from different surfaces, like from the floor and so on, and we were interested what kind of pathogenic bacteria could survive. So we identified we have to focus on the escape pathogens, which are listed here, and they are known as let's say escape pathogens because they can evade the effects of multiple antibiotics, and their control requires strict infection prevention, the hygiene, and antimicrobial stewardship as well. So we turned into that direction in for the third part, and we took the samples as it's before, and after when we inoculate the samples in a half concentrated TSB broth, and in some cases some different enrichment broths. For example, in case of Staphylococcus aureus, we move forward with the detection. And next slide, please. And what we figured out from the samples, before the disinfection, almost all of them were found in the samples, but after the counterflow treatment, only a few remained, mainly from the drains and the specific surfaces. And what we can, of course, the bacillus, it's a spore forming, it wasn't in escape category, but we could find it after the disinfection. And the most important was we found a Staphylococcus aureus from a really strange surfaces, because I don't think so it should be there, but it was there. But after the disinfection, it's eliminated. And it's also really important, the Aromonas and the Pseudomonas could find after the disinfection from the drain, of course, and some specific surfaces. So it also was a nice result to see we can eliminate the escaping categories, of course, but we have to perform really well the cleaning, and after we have to perform the disinfection, but the counterflow can help us in that challenge. The next slide, please. So what was our conclusion in the last 2 years with the Equine Clinic? The staff was satisfied, and I think it's a great news. The education and the training the staff is essential, as it figured out, and it's really essential. It's also really important, the antimicrobial resistance in the clinics on the farms. It's maybe a next project or the next chapter in our lives, but it's really important because we could detect some bacteria which can have that antimicrobial resistance, and maybe they can transfer it. It was also obvious the potentially pathogenic bacteria are also on the farms and the clinics, as of course. So their control is really important. And it's also figured out the water and the air are still an important route for the bacteria and the microbes as well. So I think that was my last slide. Yeah, so thank you for your attention.
Thank you, Tekla. Just in time, so that's perfect. Thanks, Kedal. Is there any question? Okay, so if
thank you very much, Jose Luis.
Thank you, Tekla. Thank you very much. I think that it has been very interesting. Well, I will have some questions, but I will ask you later. But
yeah, but we can discuss it in later. Of course.
Yeah, for sure. Thank you very much.
Yeah, and actually we have lots of questions because if you have a project and the experiments, it's always raised dozens of questions.
Yeah, for sure, for sure. Thank you so much, Tekla. So let's move to the third presentation of this webinar, that is from Rafal Gorny, comparison of natural biocides in an aerosol chamber with different microorganisms. Rafal Gorny from the CIOP, that is the Labor Health and Safety Institute
Central Institute for Labor Protection, National Research Institute in Poland. So I can help you translate this abbreviation.
Perfect, perfect.
I try to start my presentation and just share my screen. If I will be not successful, I will ask Christina to help me, but let me try first.
I think you can share because I granted you as presenter.
Okay. Let me try to add the right file. Hopefully it would work. Biocide. Okay. It's downloading. Please wait.
We are patient.
Yeah, 25%. Still 75 before us. Okay, that's not depends on me, but depends on the system. So hopefully it will appear soon.
That's the time when
I'm in the middle of wave, 50%. So it should be immediately.
That's the time when Windows asks for patience.
Yeah, 65, 70, 80. We're almost done. 95, 100, and converting. Still converting.
Lent is a time for conversion.
It's still, moving around. So conversion still. Okay, not waste time. So Christina can help me with that. So, finally, well, yeah, it's visible. Thank you so much. My computer, cooperate with me. So let me briefly introduce the results from the work package 2, task 2.3, which was titled Room Lab Test of Low Toxicity Biocides and Dynamic Aggregation Technologies to Aerosol of Microbial Models. It was experiments which were done in March last year in our lab in Warsaw at Central Institute for Labor Protection. It was joint efforts, so except my team from Warsaw, they joined us Christina Alamo, which represented University of Alcalá, and Sonia Peiro, representing Counterfog, and all this team you can see right now on the screen. All these people produce these results. So how we did these experiments? So we tried to generate and use our experimental setup, which was built from aerosolization chamber. Aerosolization chamber was placed in biosafety cabinet, BSL2. So what we have inside, we have two devices which were able to generate aerosols. One generated tested microorganisms. In our experience, we tested 4 different microorganisms representing 4 different groups. It was Staphylococcus aureus representing gram-positive cocci, Escherichia coli representing gram-negative rods. This is 2 microorganisms, what Tekla already mentioned, was from the group from escapee microorganisms. And we added something which representing 2 different groups. I mean, filamentous fungi, which we called molds. It was Aspergillus versicolor. And as a virus model, we used bacteriophage FX174. This bacteriophage, it is a good surrogate in biological studies representing SARS-CoV-2 models. So we try to generate, first of all, at the beginning of experiment, the aerosol, monobioaerosol of this 4 separate microorganisms. I mean, Staphylococcus aureus, Escherichia coli, Aspergillus, and bacteriophage as a virus. This generation was duration for 5 minutes. And then after 5 minutes, we generate additional aerosol. It was low toxicity biocide. After 5 minutes generation, for additional 5 minutes, we mix these 2 aerosols. And after that, we sampled for 5 minutes using filter sampler, the sample of this aerosol, and we checked viability of microorganisms after this mixing. So how the results were look like? For the we tested 4 different biocides. I forgot to mention that. These 4 different biocides was 2 natural and let's say 2 chemical biocides. Natural representing by emulsions of oregano oil and lemongrass oil, and these 2 chemicals was hydrogen peroxide, 8% concentration, and hypochlorous acid. So how the results looks like? After the generation, we got mixed results. So for these natural biocides like oregano or lemongrass, we observed that in majority of cases, so the decrease of the concentration of viable particles, microbial particles, was not, let's say, not quite visible. Some of these biocides even support the growth of microorganisms. So they were not much efficient in terms of decrease of the number of viable cells. For the chemicals, we have different situations. So hydrogen peroxide and hypochlorous acid was quite efficient and decreased the concentration. In some cases, like for the hydrogen peroxide, for example, for Escherichia coli, so they even decreased till the 0 level, so they destroyed all viable bacterial cells. For the hypochlorous acid, this effect was even more pronounced, like you see on the right screen of this picture. So for Staphylococcus aureus, Escherichia coli, Aspergillus versicolor, they practically disappear as a viable cells from the air. And for the virus, this decrease was quite substantial and decreased substantially the concentration of viable cells. If you want to control this on the plate, there's few examples how the results looks like on the plates. So before the generation with different dilution, this is control samples of the upper rows, and after mixing with the biocide in the lower parts. Like you see from the organic biocides, like biocides from oregano and from lemongrass, this is decrease of the concentration was not quite visible. But different story when you are mixing these aerosols, bioaerosols with some chemicals which we tested, like hydrogen peroxide or hypochlorous acid. You can see that really some colonies are disappear from the environment. The same was characteristic for bacteriophage. Like you see, for control samples, we have some colonies, and chemical biocides were quite efficient and decreased the number of colonies. But for natural biocides, this decrease was not visible. And like I mentioned before, we observed even some support for the growth of the colonies after that. So how we can conclude it? Definitely, chemical biocides are more efficient than these natural biocides. Because hypochlorous acid was quite efficient, we tried to test a different concentration of this low toxicity biocide, and we tested hypochlorous acid at 2000 ppm concentration and 500 ppm concentration. And like you see, concentration means something for the efficiency of the removal. So if you have less concentration hypochlorous acid, some strains like fungal strains can survive this exposure. But high concentration, up to 2000 ppm, was quite efficient, and only bacteriophage decreased his concentration but was able to survive. Any bacteria and any fungal tested in our studies was not able to survive this exposure. So for the conclusion, we can say that oregano promotes the survival of bacteria and viruses, slightly reduce the survival of molds. Lemongrass does not reduce the survival of Staphylococcus aureus and Aspergillus versicolor, slightly reduce survival of Escherichia coli, and even promotes the survival of viruses. Hydrogen peroxide significantly reduced the survival of Staphylococcus aureus and mold, and also viruses, and practically eliminates viable Escherichia coli cells. And hypochlorous acid at concentration of 2000 ppm completely eliminates viable particles of bacteria and fungi from the air and significantly reduce the survival of viruses. And less concentrated hypochlorous acid completely eliminates viable bacterial cells, significantly reduce the survival of mold and virus. So we can conclude that probably from our experiments, hypochlorous acid at concentration of 2000 ppm is the most effective agent in reducing microbial contamination, and can be advised to for example, farm owners just to use it, just to clean its surfaces in the. So that's the main conclusion from our results. So thank you very much for your attention, and I'm passing the voice to the Jose Luis. Thank you.
Thank you, Rafal, for these interesting results. And yeah, it's quite interesting that hypochlorous acid can kill all bacteria and fungi, but indeed viruses are really tough with.
However, the decrease in concentration was visible, so that's the best solution which we tested. So that's really works for all kinds of microorganisms which we tested. I mean, bacteria, fungi, and example of the virus.
Yeah, the interesting thing is that it's quantified. Let's say you have the number of logarithms that it has reduced. So it's that's the interesting part. So if there is any other question. Okay, I want to thank you for fitting to the time schedule that we have, and I will then remind you what is the next webinar that we have. Okay, so no, this is the current one that we have finished, and this is the next one. The next one will be at 12:30. We will have now half an hour for a break, and then the next webinar will be at 12:30, starting with Antonio Alcamí from CSIC. Then we'll have Diana Yorga's presentation on standardization, and later we'll have the presentation of Sonia Peiró for the contamination and prevention of pathogen transmissions in vehicles and persons. So, well, we will see you later then. Thank you. See you.
Thank you. See you in half an hour. Thank you.